Right, that brings me on to our next session on traditional Japanese beverages. So I have joining us just now the FEMS Ambassador to Japan, Ken-ichi Yoshida, and Hiroshi Takagi. Hey, we have you both. Let me bring you on.
Hello, Joe. How are you doing?
Hello, how are you both doing? Konnichiwa. Konnichiwa. And it's fantastic to have you in the room today. We're right on time. So, Ken, shall I hand over to you to be our master of ceremony?
Thank you. Thank you very much.
I can't wait to learn about traditional Japanese beverages. I'll be here in the background. Send me a chat if you need me for anything. But I'll be watching and enjoying the presentation. So thank you for joining us. And over to the both of you. Happy International Microorganism Day.
Thanks. All right. So we get started by watching introductory film. Let me share the film. Hi there. I'm now visiting Kikumasabune Sake Brewery Museum today. I'm going to introduce you how Japanese sake is made in traditional way. Let's get started. This is a model of a dormitory of sake producing workers. Most of the workers in sake brewery are originally farmers, and in the winter time they can make sake coming here. However, this is not their originally inhabited place, so they produce this dormitory to stay together and work together. This is a water mill. They had this water mill to polish the rice. There used to be numerous water mills, something like this, along the stream. As you may know, sake is produced from rice. But the original rice has a contaminant on the surface. That is why the rice has to be polished enough to produce the core part of the grain. You see, gradually the rice can turn into whitish. This is a removal of 30% of the surface. This is a removal of 40%. And finally, this is 60%. And higher purity of rice is promising to have a better quality of sake. Water is also important material to produce good sake. People extract the underground water by using this well. After polishing the rice is soaked in the water and steamed. Using this gigantic steamer, the rice is completely steamed. And this is a big cauldron to have hot water, and on the top of that, they place this steamer to finish the completion of the steaming of rice. Once the rice is steamed, this is covered with the koji mold, fungus. Then the fungus can penetrate into the core part of the rice. Then it can produce a lot of enzymes to degrade the starch. Then the starch is saponificated into sugar. This is the seeds for the alcohol fermentation. And to make the mold-covered rice, it takes a couple of days in a higher temperature. So that's why this area is insulated with rice straw to keep the heatness of inside. After finishing the fungus growth on the surface of rice, the rice and the fungus all together mashed in the way as you can see in the center of the television. And probably you can hear some music around. This is a song for the mashing the rice, and they can count the time as the number of songs sung along. So this is a very clever way to scale the time without clock in ancient age. And during this mashing, the sake seeds is inoculated with various microbes, including yeast and lactic acid bacteria. And lactic acid bacteria grows earlier than yeast to make the circumstances acidified, good for to avoid the contamination of other species, and better for the conditions for yeast growth. So after the mashing, the mashed rice with the fingers and additional rice, steamed rice, and water, they start alcohol fermentation in this barrel. And they increase the scale 3 times in a stepwise fashion. So 3 times means 8 times of the volume of the sake is produced in the biggest barrel over there. So the fermentation lasts about 1 month's time. And after the finishing of fermentation, the sake and the other materials, everything is packed in this bag. And this bag is made with jute. Then this jute bag is clustered or like piled up inside of this presser. And you can see the long stem here. By using this power of pressure, they press the sake fermentation. Then they can extract a clarified sake underneath like this. And after completion of the pressing and filtrating, they keep the fresh sake for half a year time in this big barrel. You can see the seals on the top of the barrels. This is to stop the oxidation further. And after that, 1.5 year time, the sake is macerated and to be ready for the market service. Yeah, these are the selection of fine sake glasses. You can see most of them are made ceramics, and such a beauty they enjoy. And actually, this is a good relation of sake drinking here in Japan. And there are some other variations made with wood or bamboo, and also funny faces also written there. So the equipment which was used in the ancient age, especially for some ceremonial moment, they served the sake as a kind of gift from God, and that is why things are more has a flavor of a traditional relationship. You can see they recorded it beautifully.
Me de ta no na wakamatsu samayo
I hope you enjoy the museum and also the technology and history of making sake. I hope you can have a chance to enjoy Japanese sake in reality, and I hope you will like it. Thank you very much for your kind attention. So, hello again, everybody. Good morning, good afternoon, or good evening, whichever, depending upon where you are. And welcome to this session to talk about microbiology and traditional Japanese alcoholic beverages. And my name is Ken Yoshida.
I am the FEMS Ambassador to Japan, and it's my great pleasure to moderate this session. And I hope you enjoyed the introductory film in which I introduced to you the traditional Japanese sake brewery museum. And probably you have already some questions, and don't be hesitate to post your questions into the chat. And actually Joe is in the background to collect all the questions, and so that we try to answer them as much as we can later. Actually, you are responsible to make this session interactive, right? And not only that, today we have the specialist who can provide more precise information about the Japanese beverages. And let me introduce you Professor Hiroshi Takagi from Nara Institute of Science and Technology, Japan. So good afternoon, Professor Takagi.
Good afternoon.
You're right. Yeah, so
Yeah.
could you please introduce yourself by using your slide?
Mm-hmm. Can I share the screen? Yeah.
Yes, yes, please.
Can you see?
Yes, here you go.
Okay. Can you see the screen and my voice? Can you hear my voice? Okay.
Yes,
Yeah.
everything's okay.
Okay. Yeah. Ken, thank you for your kind of introduction about me. So good morning in European, and good afternoon in Asian, and also good evening in the United States. It depends on the place where you join this program. So my name is Hiroshi Takagi, working at historical Nara, but living in also historical Kyoto, Japan. So my lab is involved in the applied microbiology that covers the basic studies in microbial science and the practical application in biotechnology. So today, I'm greatly honored to be here with Ken. And now we will talk about the traditional Japanese alcohol beverage brewed with lovely microorganisms. Okay, so until last month, I was the president of International Commission on Yeast, ICY, which is under the umbrella of the International Union of Microbiological Science Societies, IUMS, since back to 2016, in which I organized a General Congress ICY 14 in Japan. So this is Isabel. She's a great Portuguese yeast scientist and kindly invited me the last year as the speaker of IMD 2020. Okay, personally, I like any alcohol beverages very much. So there are two types of drinks as shown here. So non-distilled alcoholic beverages such as beer and wine, and distilled spirits such as whiskey and brandy. So among them, so sake and shochu, including Awamori, are very unique Japanese products by excellent collaboration with two kinds of microbes, yeast and fungi. So basically, the alcoholic beverages contain the ethanol, which are mainly produced by the fermentation, that is the metabolism of the carbohydrates by yeast under the anaerobic or low oxygen conditions. So this Latin word means boil in English, that to emit or to release small bubbles of gas, carbon dioxide, as carbonated or fermented liquid. Also, not only alcoholic beverages such as beer, wine, sake, but also bread and bioethanol normally use yeast cells at some stage of the production. So in the case of the alcoholic beverages, the process is called brewing. Okay, so one of my research project is involved in the tolerance to the high concentration of ethanol that is very toxic to cells, these cells. But I am very sensitive to the high concentration of ethanol when I drink too much alcohol beverage at night. Okay.
Yeah, Professor Takagi, obviously you love alcohol drink.
Yes.
That probably this is the
You
reason
too.
you're a specialist in the yeast nowadays, right?
Mhm. Yeah.
So, but anyway, and obviously, to produce alcoholic beverages, yeast is the main player. So,
Mhm.
could you please explain us how it works for alcohol production?
Oh, yeah. Yes, I will continue to answer your questions. Okay. So I will briefly show you the yeast Saccharomyces cerevisiae. This is an important microorganism in basic science as a model for higher eukaryotes. The past, the Nobel Prize, the winner, Professor Yoshinori Osumi, discovered and elucidated the mechanism for autophagy, which is a fundamental processes for degrading and recycling the cellular components. Also, yeast is a useful microbe in fermentation industry due to the extremely high ethanol productivity. For example, as I will talk about later, I isolated yeast mutant that overproduces isoamyl acetate, known as the key flavor. So by brewing with my yeast, Awamori, which is a traditional distilled alcohol beverage in Japan, has been successfully commercialized. Okay, so one of the alcoholic beverages which we focus on today is sake, called the Japanese rice wine. It is historically said that the Shoryakuji in Nara or the Konoike in Itami, Hyogo Prefecture, both of which are not so far each other, is the birthplace of sake. So since sake is an integral part of the Japanese culture and the lifestyle, it has been approved as the national alcoholic beverage, kokushu. Okay. Also, Japanese cuisine is becoming the more popular internationally, so the increasing number of people enjoy sake with the Japanese food all over the world. It is basically labeled with the word seishu, refined alcohol. So sake is made from the steamed rice. By the multiple parallel fermentation of the fungus Aspergillus oryzae and the yeast Saccharomyces cerevisiae, which produces the saccharification enzyme and ethanol from the glucose, respectively. Now, let me introduce more details how to make sake. Sake is made from the rice that has been polished to remove the bran by the very good collaboration of the yellow koji Aspergillus oryzae and the budding yeast Saccharomyces cerevisiae. Rice starch is degraded into sugars catalyzed by the hydrolyzing enzyme from the Aspergillus oryzae. After that, the Saccharomyces cerevisiae converts sugars into the ethanol by the glycolysis and alcohol fermentation. So, okay. So the yeast cells produce not only ethanol, but also the aroma and flavor compounds. Both of them are very important sake components. And this alcoholic fermentation is required for the sake making, as well as the production of the wine, beer, the bioethanol, and also bread, as you know well. Okay, so I would like to also explain the rice for sake brewing. So the surface of rice contains a lot of the nutrients such as protein and fat. In the sake brewing, it is necessary to polish off the surface of the rice because these elements can cause an unpleasant taste. So this graph shows the changes in the proportions of the components caused by rice polishing. The table rice, which we normally eat, has the rice polishing ratio of about 90%. This means the 10% of the extract of the grain was removed as brown. So we consume the remaining 90% as rice. So especially the designated, the sakes are classified according to the rice polishing ratio. So for example, the Daiginjo sake is clean and less complex because it is made from the highly polished rice, but is very expensive because it requires a large amount of rice as it is polished a lot. And Ginjo sake is usually fruity and fragrant with the rich flavor because the rice is less polished. But I'm not saying that the high price is the best. So your taste will be able to judge or decide the best sake for you. Okay, so regarding the fungus, Aspergillus oryzae, recently the gene expression of the Daiginjo koji and also Junmai koji was comprehensively compared. So the result clearly shows that there are differences in the expression for about 3,500 genes. So more importantly, the many genes involved in the amino acid metabolism were highly expressed in Daiginjo koji. In contrast, the Junmai koji nicely expressed a lot of genes involved in the lipid metabolism, leading to the flavor making. So rice polishing ratio that greatly affects the expression of the fungus genes, contributing to the diversity of taste and flavor in sake. Okay, do you understand?
Thank you. Amazing, because this sake production involves many different organisms, mainly the yeast and fungus, koji mold, right?
Yes.
So actually, it is amazing that these a lot of different organisms are controlled to be a good result, to birth a good result of fermentation. It's really amazing, and actually, all those microbes are collaborating in a good harmony. So that is why I think every single time I enjoy sake, it tastes like a good fragrance and alcohol in a good harmony. Yeah, it tells the way as exactly the nature of its beverage taste. All right, so you mentioned about the Daiginjo sake, most fragrant and expensive ones. And I got another film from the producers in Kobe, which is called Hakutsuru Shuzo. And we can see how nowadays they use all those microbes to make the sake. And let's see this video together now, about 10 minutes time.
当社アクツル酒造の大吟醸作りについてご紹介します。まずはじめにゼンマイ工程です。白米は袋に正確に10キロずつ計量して分けておきます。大吟醸に使用する米は 38% まで削った酒米の王者と呼ばれる山田錦を使用します。 10度の水を張った半切れ桶につけて3拍子のリズムで手洗いしていきます。手洗いは1分間を2回、時間は正確にストップウォッチで測ります。ピッピッと聞こえるリズムは3拍子のメトロノームの音です。洗い手によって洗い方が変化しないようにリズムを合わせます。2回の手洗いが終わると1分間シャワーで汚れやぬかを洗い流します。10度の水が入った半切れ桶に米をつけて7分から13分浸漬します。多く吸水させてしまうと酒造りに悪影響をもたらすため、非常に厳密に管理します。浸漬が終わると脱水機で30秒間脱水します。この作業を一仕込みでは68回行うことになります。脱水後、吸水具合を測定します。この時の吸水具合は 131.4% になりました。目標吸水具合は 131.5% なので非常に近い値となりました。翌日は米を蒸す作業です。翌朝、古式に米を入れていきます。蒸気を入れ、米を蒸していきます。酒造りでは米は炊くのではなく蒸します。1時間蒸し、最後に5分ほどスーパーヒーターを入れることもあります。蒸し終わると少しずつ運び出し、計量してから床の上の麻布の上にさらしていきます。2人組で粗熱を取ります。40度程度になったところで麹室に取り込んでいきます。床の上で広げていきます。2時間ほど冷まして品温が32度、水分が 34% 以下ぐらいに下がったところで種を切ります。種切りはもやしが上から巻き上がるように振ります。底もみをした後、水分蒸発による品温低下を防ぐために布でくるんでいきます。量が少ないのは元麹であるためです。くるんだ後の温度は 31.4 度。翌朝、温度が2度以上上がっていれば塊をほぐす切り返しをします。当社では人数をかけて短時間で終わらせる方法を採用しています。2人で1時間かかる作業を6人で20分で終わらせます。こちらが切り返しの時の麹です。ふるいにかけて米粒に分かれたものを集めていきます。麹蓋に麹を盛っていきます。5枚積んだ上に空蓋を2枚乗せ、その上に布をかけて保温します。これは急激な品温上昇を抑えるためです。34度で1回目の積み替えをし、36度で中仕事です。麹を手でよく混ぜます。中央に盛り上げ、真ん中に くぼみを作ります。最後にハケで寄せます。38度で2回目の積み替えをし、40度を超えたところでしまい仕事です。麹を手でよく混ぜます。中央に盛り上げ、2列の線を入れ、小判状に広げます。夜中に2回、午前中に2回ほど積み替えをし、手麹です。手麹のタイミングは、麹力か、溶合、香り、甘みなどで判断します。この時の麹は栗のような香りがします。麹を広げて冷まします。次に仕込み作業に入ります。まずは元の水麹です。タンクに麹を入れます。仕込み水を入れます。ハイボーで攪拌します。次に吟醸酵母を入れます。吟醸酵母は他の酵母と比較して高濃度のアルコールを作り、果実のような香りを出すのが特徴です。翌朝、蒸し米を入れます。よくほぐしながら入れていきます。仕込みはまず、酒の元となる酒母を作り、その酒母をベースとし、添え、中、染めと3回に分けて原料を添加していきます。こちらは最後の染め仕込みの様子です。2800リットルのタンクにもろみ量は1500リットルぐらいです。染め仕込みの温度は6度程度です。もろみの情報を見ていきます。3日目はまだ流動が始まっていません。5日目です。米が動き出し、泡が出始めています。7日目では大小の泡がたくさん出ています。10日目です。このあたりで吟醸酒特有のリンゴのような香り、カプロン酸エチルを酵母が生産します。15日目、発酵がだんだん落ち着いてきました。25日目、上に蓋のようなものができています。33日目にようやく醸造です。ここからお酒を醸造します。これは袋釣りと呼ばれる方法です。もろみを酒袋に入れます。金具をつけてタンクに吊るします。1本のタンクに30から33本の袋を吊るします。荒走りの濁った部分を弾きます。成長になったところを摘出します。ここが一番楽しみで、そして怖い瞬間です。問題がなければ土瓶に取っていきます。約1500リットルのうち80リットルのみです。こちらが新酒となります。
Right. It is amazing. Everything is so precisely controlled. And so they did a lot of manufacturing. This is the good way of conditioning the growth of fungus, isn't it? It's very amazing. And so, Professor, they say that they use ginjo yeast. This could be a very special species, I think. And how they choose or how they breed this kind of specific yeast. Could you please introduce how microbiologists are contributing to this sake fermentation or sake brewing, please?
Okay. Can you share? Can you see
Yes,
my?
yes, please.
Yeah.
Yes, go ahead.
Okay. Yeah. Well, first of all, I will briefly show you how to construct the commercial brewing yeast. For the application of the recombinant yeast, GM yeast, genetically modified yeast, and the self-cloning yeast, which contains no foreign genes or sequences except for yeast DNA, is more acceptable for consumers than GM yeast. According to the Japanese government guideline, the self-cloning yeast does not have to be treated as GM yeast because self-cloning processes are considered to be almost the same as naturally occurring gene conversions. However, the Japanese food industry has not yet accepted self-cloning yeast. Okay. Wait.
Yes, so it seems like
Oh, yeah. Okay, yeah.
natural
So, yeah,
selection
I'm
is
sorry. Yeah.
required, yeah?
Right. So we construct the brewing yeast strains by the conventional mutagenesis, which is a traditional and widely acceptable method for breeding. They are in food industries and also for consumers. Okay, so as an example, I am very happy to introduce you my recent research topic about the healthy image sake with the high level of ornithine, one of the amino acids that is one of the functional amino acids. So this is a collaboration with the R&D Center of Nara, where is said to be the birthplace of Japanese sake, and Bodai Moto, that is a prototype of rare raw sake. So this is a unique method of the killing various bacteria with the lactic acid produced by the lactic acid bacteria before the yeast becomes active. So we recently isolated the interesting yeast mutant that accumulate amino acid protein and ornithine in the cell. So this ornithine is a non-proteinogenic amino acid, which with the physiological functions such as reducing fatigue or improved sleep quality. So high amount of ornithine they're contained in shijimi clam and shimeji mushroom. So regarding this interesting mutant, we identified a mutation in the ARG6 genes. They're encoding the threonine 340 isoleucine variant of the N-acetylglutamate kinase, which is insensitive to the feedback inhibition by arginine, so leading to the ornithine accumulation in the cell. So finally, we brewed laboratory scale sake and analyzed the fermentation profiles of the parent strain and this mutant strain. So after fermentation, the sake and the sake cake, sake kasu, brewed with this mutant contain the large amount of ornithine relative to the dose brewed with the parent strain. So recently, that we successfully commercialized this healthy image sake containing a high level of ornithine by local 3 breweries in Nara Prefecture. Okay, that's all about sake.
Wow, that's amazing. Because as you said, that Ornithine is specific for freshwater crumb, right? So then this sake tastes like freshwater crumb?
Yeah, but just a healthy image, because too much drinking sake is not so good.
All right, but actually Ornithine is good for health in liver.
Right,
So
right.
consuming a
Yeah,
lot
so
of
before
alcohol
and
requires
after
probably
drinking
the
too much,
liver.
yeah. Normally we have to purchase the supplement of origin, but you don't need.
Yeah, so it sounds like this is a good compensation of drinking a lot of sake. Great
Yes.
thing. It's quite funny.
Thank
And
you.
but actually, it is impressive that in the behind of this sake brewery, actually, you scientists are working to produce better function in the yeast. So this is very impressive. Thank you,
Right.
Professor. So by the way, I have heard like there is another version of sake which is distilled, and maybe you mentioned about Awamori, is that right? So
Right.
could you please introduce it for us?
Yes. Thank you very much once again. Okay, so the Awamori, one type of shochu is the distilled spirits. They're in the subtropical islands, Okinawa, which is the southernmost and the westernmost prefecture of Japan. So Okinawan cultures that bears the trace of the various trading countries, such as China and Thailand, so which influences the island's culture, customs. Okay, so it is said the name of Awamori there is derived from the phenomenon that the form, the awa in Japanese, would rise up the mori in Japanese during the distillation. So basically, the Awamori is made from the indica rice exported from Thailand by the multiple powder fermentations of the fungus, another fungus, Aspergillus luchuensis, and the yeast Saccharomyces cerevisiae. So which produce the saccharification enzyme and also the ethanol from the glucose respectively. So after the fermentation, the alcohol is normally or mainly distilled and aged. So now let me introduce more details how to make Awamori. Its brewing processes is quite different from that of other distilled spirits like whiskey. So made from the grain mash, where the conversion from starch to alcohol via sugar by malt enzymes and yeast. So during Awamori brewing, the fungus Aspergillus luchuensis and the yeast Saccharomyces cerevisiae are doing very good job for degrading rice starch into the sugars catalyzed by the hydrolyzing enzyme for preparing the fermentation mash, moromi, and converting sugars into the ethanol by the glycolysis and alcoholic fermentation, respectively. By the multiple, of course, parallel fermentation, it means the simultaneous, the saccharification and fermentation. So after the fermentation, the alcohol is distilled under the atmospheric pressure and then is put in the barrel or a barrel for aging that develops rich and strong flavors. Okay? So when Awamori is aged underground in the constant cool temperature in the clay pot or vases for 3 years or more, it is called kusu, the old liquor.
Right. So we got another film from Awamori producer, so we can see it now.
沖縄を代表するお酒、泡盛。何世紀も世の人々を魅了し続けるその芳醇な味わい。泡盛は15世紀半ば頃、東南アジアや中国から伝わったとされる日本で最も古い歴史を持つ蒸留酒です。琉球王朝時代には王府の御用酒として珍重されました。上村酒造は明治15年、那覇市半田川において上村清心が創業し、1世紀余りにわたって培われてきた手作り製法にこだわり続ける泡盛メーカーです。泡盛の原料には主に硬質のインディカ米を使用します。まずぬかを取り除くために原料米を洗います。次に水をため、蒸すために必要な水分を原料米に浸積させます。浸積した原料米を十分に水切りした後、蒸気で蒸し上げます。蒸すことにより麹を作りやすい状態にします。蒸し上がった原料米を適温まで冷まし、黒麹菌を散布します。この黒麹菌を使っていることが泡盛の特徴の一つです。黒麹菌を散布した原料米を生菊棚に移し、温度管理をしながら麹の育成を図ります。黒麹菌の生育により米のでんぷんをブドウ糖へと変化させる酵素を作り上げます。この時クエン酸も生成され、より良い泡盛作りの環境を作ります。このように全体に黒麹菌をつけた原料米を米麹と言います。米麹に水、酵母を足してもろみを作ります。このように米麹と水だけを原料に一度に仕込む全麹仕込みが泡盛ならではの特徴なのです。麹から出てきた糖分を酵母が食べてアルコールと炭酸ガスが生まれます。2週間ほど経つとアルコール分 18% 前後のもろみが出来上がります。出来上がったもろみを蒸留機に移して蒸留します。蒸留は水より低い温度で蒸発するアルコールの性質を利用して純度の高いアルコールを取り出す技術です。蒸留して生まれた泡盛は亀やタンク、オークダルなどに入れられて貯蔵されます。蒸留してから半年ほど経つと一般酒として瓶詰めされますが、古酒にするには最低でも3年以上の熟成が必要とされます。このような過程を経て泡盛はボトルや壺などに詰められて出荷されます。当社の主な商品としては、伝統的な泡盛の風味を程よく引き出した酒霊と、甘い香りと豊かなコクを特徴とする琥珀色の樽貯蔵泡盛暖流、そして上村酒造独自の芳醇酵母で作った原酒51度や香ばしくて旨味の濃い泡盛芳醇ロマンがあります。これらの銘柄は国内外でも高い評価を受けています。酒霊はこれまでに泡盛鑑評会において県知事賞や優等賞を数多く受賞。暖流は全国酒類コンクールやベルギーで行われている国際品評会モンドセレクション、 ITQI 国際味覚審査機構において高い品質を認められております。芳醇ロマンにおいても全国酒類コンクールで1位を受賞するなど高い評価をいただきました。いずれも自信を持ってお勧めできる泡盛です。 青森のタルチョ像は当社が最初に研究開発に取り組み商品化に成功したことで知られています。日本で唯一地上戦が行われた沖縄では青森のほとんどが消失しました。戦後上村酒造は官営工場として製造を再開。原料も限られたものしか手に入らない時代からより美味しい青森を提供するために試行錯誤の末、当時よく飲まれていたウイスキー作りの製法にヒントを得て作り上げたのがタルチョ像青森暖流です。上村酒造では琉球時代から受け継がれている空須文化を後世に受け継ぐため、杜氏が厳選した青森を熟成に適した近蔵でお預かりしております。お客様の未来へのメッセージが添えられた青森は近蔵で静かに時を重ねていきます。上村酒造では澄んだ空気と豊かな緑の中でこれからも受け継がれた技と心を育んでまいります。
Right, that's great. Obviously, the black koji, kuro koji, is the specific for
Yes,
Awamori
yes.
production. Could you please explain why they chose this black koji for Awamori production?
Okay. Yes, just a moment.
Maybe they produce a specific compound for the purpose.
Yes, I'm really sorry, just that my computer is getting. Okay, yeah. Yes, I'm really sorry. Yeah. Okay, so the Aspergillus luchuensis is cultivated on steamer rice under the suitable conditions for the growth. The resulting kuro koji, black koji, contains not only the wide variety of enzymes, but also citric acid that protects against spoilage during the fermentation. Okay. And as I mentioned, the Awamori becomes the more freezed after aging due to the increase in the mellowness and flavor. So one of the components, I'm really sorry. Okay. Well, yeah, I'm sorry. So one of the contributing compounds is vanillin. Okay, the vanillin, the starting material is the ferric acid, attached on the cell wall polysaccharides of the rice grain. So the enzyme ferric acid esterase produced by the black koji can release the ferric acid into the monomer. So free ferric acid is converted into the 4-vinyl glycol, mostly by the enzyme ferric acid decarboxylase produced by the certain microbe in Awamori. So finally, 4-BZ is transferred into Awamori in distillation and is then chemically converted into the vanillin. This is the key flavor for aged Awamori.
I see. So it means like this has a very sweet fragrance because of
yes,
vanillin.
So there are,
okay,
yeah,
great.
Very good job.
Yeah, it is. And Professor, I've heard you have your own production of a specific brand of Awamori. Could you introduce what is the difference from others?
Yeah. Okay,
Just
so
briefly.
finally, also it is my great pleasure to show you my recent research topic about aromatic awamori with the high level of isoamyl acetate. That is one of the key flavor for alcoholic beverages. So this is a collaboration with the bioventure company Biojet, which isolated the novel Saccharomyces cerevisiae from the hibiscus flower in Okinawa that can brew awamori with the high level of ethanol. And sometimes okay, so isoamyl acetates here is the known as the banana-like flavor, and also the key flavor ginjoko in Japanese sake. So in yeast, isoamyl acetate is converted from the alpha keto isocaproate, which is an intermediate of the amino acid leucine pathway. So in the three steps catalyzed by these enzyme, finally the isoamyl acetate formation. So the isoamyl propiate synthesis, IPMS, this is the key enzyme that controls the leucine synthesis. Because this enzymatic activity is subjected to the feedback inhibition by leucine. So the hibiscus yeast produces the high level of ethanol, but the isoamyl acetate content is quite low than observed in the classical Awamori yeast. So to increase the isoamyl acetate content, we isolated the interesting mutant with the leucine accumulation in the cell. So regarding in this mutant, we identified the mutation in the enzymatic gene encoding the variant of this enzyme. So which is insensitive to the feedback inhibition, leading to the leucine and also isoamyl acetate accumulation in the cell. So finally, finally, in the laboratory scale, awamori brewed with this mutant. You can see the very good job, including leucine accumulation and also isoamyl acetation in awamori compared with the parent strain. So so many times traveling. Okay, so finally, we recently commercialized the two types of awamori brewed with this mutant. So in Okinawa Island.
Wow, brilliant! Yeah. I'd like to have one day this your vanilla banana mixture of awamori to be lovely. Right. Thanks. So maybe we can have 2 minutes for a Q&A session. And there are some questions. Okay, so we can say the different tastes in the sake. So I think if we have residual sugar, the taste could be sweeter, and alcohol is higher, it can be drier, and sometimes we can have acid, it can be sour. And also it's been told like we have a fruitish flavor. So these balancing make the different taste. So you can be very choosy to have good sake.
Right. So wide variety of sake in Japan.
Yes. And another thing is you have
this is kind of the evidence for my research.
Very good. And there's one funny question. Do you think it is possible to produce beer using sake yeast? It's known, or it's good or bad?
Okay, basically saying no, because the beer yeast is required for the utilizing the maltose. Okay, so basically the sake yeast and the awamori yeast is not so good for maltose utilization. So beer yeast, for beer yeast, we should use the maltose utilizing yeast.
I see. So everybody, beer is beer, sake is sake. We have to make it discriminated. All right, good. So time is up. Thank you very much,
Mhm.
Professor. We enjoyed a lot. And everybody, you can have a chance to enjoy Japanese alcoholic beverages. You must get it at least once upon a lifetime. Okay, Joe, we made it.
Hello, thank you so much.
Thank you.
And I really do hope I get to enjoy sake. I mean, it's 10 a.m. here, so maybe not right now, but maybe later today. Yeah, there we go. I hope we can get some somewhere in the Netherlands. I had one more question for you. And that's, what is the best way to drink Awamori?
Yeah,
Oh,
Professor, you know it.
yeah. Basically, in Okinawa Island is the subtropical region. So I recommend the water, with the ice.
Yeah, okay. So a bit of ice, a bit of water.
Not with
Nice.
hot water. Yeah.
Hot water.
No.
Yeah,
I don't
no.
recommend. Yeah.
So ice, cold water,
Ice, water.
a
Yeah.
nice cup, and then we sip it.
Yes.
Wonderful. Okay. Thank you both for joining us on International Microorganism Day. You've actually almost had the whole of your International Microorganism Day in Japan. We're just beginning ours. So I hope you celebrate well tonight and have the best day. And yeah, thank you so much for coming and telling us all about these different Japanese beverages. We've learned so much, so it's been very wonderful. So I bid you goodbye, and hopefully we'll see you next year.
Yeah,
Thank you very much.
Yeah. Hopefully, yes. Yeah. Thank you.
And
Have a nice rest of time.
we will. Bye, Ken.
enjoy
Bye,
still.
Hiroshi.
Bye.
Good to see you both.